Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, wassalatu wassalamu ala rasulillah, amma ba'du. Kaifa halukum? Alhamdulillah, khair ya. Are you ready, student, today? Okay, before we begin, I think we need to pray to Allah Subhanahu wa Ta'ala. So, Kati, class leader, can you mungkin your friend like me as usually to lead how to pray, like feel your breathing, or let's praise to Allah and blah blah blah. Okay, can come here. Okay, so guys, before we start our learning activity today, we, as usual, beg Allah for forgiveness to ease up all of our learning lessons and for Allah to bless this learning session this morning. Bismillah, prayer begins. While you are praying, don't forget to feel, to feel your life, why Allah still give us this life, and let's praise to Allah. Alhamdulillah. Let's say to Allah, Alhamdulillah to Allah. Thank you, Fatih. Okay. So before we begin, let me check your attendance first. Who is not here today? I think you all are here now. Yes. Malik, where is he? Go to toilet. Okay. Salah, still charging. Okay. Okay. Last night, of course, you have learned about the topic today. About our topic today is about the synthesize protein synthesize. Okay. I believe you have read and trying to understand it by your best. But I need you to explain. So I need you to explain to what you have learned and what you have understand about the cell, about the protein synthesis. Including transcription, translation, enzyme, amino acid, and everything you have understand. Please explain to me. Thank you. Okay, so please starting from Hatta. Okay. What we have learned last night, or just explain to us what you have understand about the cell, synthesis, protein synthesis, others.
Last night we already learned about cell. One of the topics was about cell. So the cell is divided into two, prokaryotic and also eukaryotic. And last night we focused more on the eukaryotic cell. So the eukaryotic cell means that the cell is there's a nucleus inside, and there's also DNA. And also we already talk about the another process like the transcription, also translation, which including the DNA. So the translation means the replication of duplicated of the DNA become the mRNA, and also the transcription when the process that translate the mRNA in the ribosome, also with the tRNA to describe to making the amino acid.
Okay, good. Next, Fatih.
Yes. So from what I've learned is that in cells they need proteins, and in order to um, satisfy that need, there is a process called protein synthesis, and within that process, there is a sequence of activities that the cell is doing, in which the first one is going to be the transcription, in which transcription translate.
Yeah,
Okay,
you made the help.
is it correct? Transcription, right?
So the first process is replicating the DNA.
In which that process is within the
No, it's different. The replicate and transcription is in different process. Replication,
the first step is transcription, right?
yeah, in protein synthesis, the first step is transcription. Yeah, replication is just to duplicate the DNA. Yeah, not for protein synthesis.
Okay, so the first process is gonna be the transcription, in which the mRNA is gonna be uh, how quote unquote copy the DNA, the original DNA within the nucleus, and then after that process, there will be tRNA, and in which this tRNA will transport the mRNA from the nucleus into the ribosome, and then after that, in the ribosome, the RNA will be translated into the gold protein. Whatever protein the cell is trying to make will be translated by the ribosome.
Good job. Okay. And to that, Fikri,
Okay, as Tati explained before, that the protein synthesis have the two stages, which are transcription and translation, and why this process is essential because protein synthesis will make a protein, and protein is as a function to be a signal for our cellular movement or others, or to be the enzyme. So protein synthesis is essential for cells, our cells movement, and how our body works. It can be controlled or can be signaled by the protein.
okay, good job.
Transcription, translation, and the last is
That's the last.
oh yeah, just two. Okay, so I'm gonna deep the conversation in the transcription section. So the transcription is the process that the DNA sequence is copying into the RNA sequence, mRNA sequence. So uh, in the DNA we have the uh, gene called AGTC, and it has its own complementary. So the A is going to be the U in the mRNA section, and the G is complementary to C, and the T is complementary to A, and the last so the G is complementary to C.
Okay, good job.
Okay, please. Have you done? So I'm gonna tell you guys about the transcription. There's three main stages from the transcription: is initiation, elongation, and then the termination. The first one, initiation. The process begins when an enzyme called RNA polymerase binds to a specific region on the DNA called the promoter. In eukaryotes, protein called transcription factors help the RNA polymerase bind to the promoter. This bind causes the DNA double helix to unwind. Second, the elongation. RNA, RNA polymerase moves one strand of the DNA, that's the template strand, and synthesizes a complementary mRNA strand. It reads the DNA base and adds the corresponding RNA base, A with U, G with C. Remember, in RNA uracil, replace the mine, yeah, it's on the mRNA, And then the termination, RNA polymerase continues until it's reaches a special sequence on the DNA called terminator. At this point, the polymerase detaches from the DNA and the newly formed mRNA strand is born.
Okay, sure. Next, Malik.
Okay, so I'm gonna talk about the DNA transcription and translation to you guys. So there is two main stages. They call the first one is called the transcription. It's like the DNA. No, it's like mRNA. Occurs in the nucleus for eukaryotes, and an enzyme called RNA polymerase binds to the DNA sequence G. Then the DNA unwinds, and RNA polymerase shifts the DNA. Translation is to translate the mRNA to the protein. Transcription occurs in nucleus for the eukaryotic organism, and translation occurs in ribosome, which is in cytoplasm.
Because we are completing each other, yeah. Inshallah, we are completing. Yeah. So now I think all are ready to learn. Yeah. So we can move to the next step. Yeah. So for today, inshallah, we will have a new goals. Yeah. What our goals today? Our goals is we will have the case. Yeah. Case study, and we will apply our knowledge to solve this problem. Yeah. And what the problem? What we have today? Yeah. So today we have the cancer patient, and this patient we need to cure. And how to cure? We need to apply our knowledge about the protein synthesis. Yeah, because the cancer cell is the what is it? There are some mistakes we need to solve in DNA sequence, right? So this we need to, yeah. In today we need to analyze, yeah, the sequence of DNA, and we need to evaluate which one better, which way better, and the last is we will creating we will create the best methods, the best enzyme, the best protein to cure this patient. But we are in our class now. Yeah, we don't have the cancer cell patient now. Yeah, and we cannot go tiba-tiba pergi keluar gitu, nggak bisa ya. So how we can do it? Yeah, that's why we have new technology. Yeah, we have MetaQuest 3 to bring us to the virtual world. And in this world, we will act like a doctor, yeah, like a scientist. Yeah, so you will solve this problem in virtual worlds. Yeah, this will simulate your skill, and maybe someday you will be a doctor, right? Yeah, you will be a real doctor, and you can apply it in your real world. Inshallah, yeah. Inshallah, yeah. Okay, let's starting by. Okay, don't forget to what is it? Yeah, don't forget to our classroom agreement. Yeah. So please, when someone talking, when someone speaking, the others should to listen. And uh, when you want to speak, what you need to do? Raise your hand. Yeah. No food, no sleep. Yeah. And respect each other. Okay. So the problem is about the what?
Cancer.
Yeah, the cancer. Yeah. Okay. First, no, I think we can use this. Also, can record ourselves using the camera in this phone. Okay. It's fine. Okay, guys. So first, we have a problem, and the problem is the cancer. We will work in teams. Yeah. So we all is one team. And Fatih, you as the class leader, you need to manage the skill of ours. Yeah. We have skill. Yeah. We have the expert, we have specialists, the other. So you need to manage it. So we will have some teams. Yeah. First, team for um, sales specialists. Yeah. Because before we can analyze, evaluate, and creating the sequence of DNA, we need to restructure the cell first, right? Yeah. So we need the cell expert. Yeah. And the second, we need to uh, transcription transcription team, and the third is about is the translation. Okay, so we will have three teams to solve this problem today. Yeah.
And has practice scripture and call it as translated because your terms are those experts in transcription and the translation one is those expert. So before I start, does anyone here really apply themselves as any one of those roles? We only serve I kind of sort of expert. Okay, doesn't be. You'd like to be what? Transcriptor, okay? Okay, I don't know the differences, but I like to be what? I write down the, wait, so can you move to here? Cell specialist, right? It's not good when you have this up there, transcriptor, transcriptor, okay? So let's start. Whether you want to be what? Transcriptor. Okay. Okay. Again, we will be something. Yes. That will be translation. Translate. Now what will do it? I think transcription is okay. So we have two transcriptions already. Available in Java. So, do you guys think two guys, two experts per role will be enough? Maybe it's okay. So I think we can conclude for transcriptors and we can only we have two roles, right? So sales pitchers and specialists. Okay. Would like to do one? Sales specialist. Okay. So that is
And I'm going to word. So specialist, okay. I thought would like to be translator because there's only one person. Okay. So now I have the name list right now of each of our all the rules. But before I, let's say, before I close this what do you call it? Registration, I would like to know you guys are expert in what you guys chose, right? Like, you don't have to be expert, but at least you know what you're doing, right? Yeah. Okay, so now I trust you guys. I trust that you guys know what you're doing and you're not just picking which one of these roles just for I'm gonna go for the easiest one. I'm just gonna pick randomly. But I trust you guys that you guys are actually knowledge in these fields. Okay, now so I agree. I think I already have the names for each one of these roles. Okay, I can put you to right here. Okay. Okay, so see for sound specialist right here, you got 3%. Okay, and that would be okay. Ali. And for transcriptures, I'm gonna put in and yeah. For translators, that's one and Hata. Okay, so we already have the teammates right here, and I think we can proceed to the next step.
Okay, thank you very much.
Now we have teams, yeah. We have a teams to solve this problem today. Our problem today. The team one is self-specialized. Who is this? Malik, Salahuddin, and Fikri. And for transcription is Beryl and Dafa, and for translation is Ezan and Hatta. So the final is problem will solve, inshallah, yeah.
Yes.
Okay, before we solve this problem, I think we need to learn again to make the good strategy, yeah. So yeah, I made the modules for this. Yeah, so you can you may to open your Chromebook and open dnaeducation.blogspot.com, and there is one new module about this. Yeah, so you can learn about it to analyze, to make a good strategy for our problem today. Yeah. So please. Are we looking around here?
Context to
You get it? Not that. Yeah, but the new one. Yeah, open it again.
oh
Yaps. That's it. Yeah, that's it. Okay, we go to our protein master, protein expert.
every code from the protein that have only specific amino acids right there.
Yep. Okay. many amino acid we have? Dafa,
Huh?
could you tell me how many amino acid
Oh,
in this
so
word?
for the example, the sequence of UUU, which means that the amino acid is called valine, and the UUA, the amino acid is called leucine.
Okay. And how many amino acid here?
30.
It's
30.
20.
20.
probably
Yeah, 20.
right, 100.
Yeah.
Okay, so there is no.
We talked about it.
Okay, everyone, Alhamdulillah, we have learned about the cell, about the protein synthesis, and today we will use our knowledge to solve the problem. And the problem is about the cancer cell, about the patient, about the human, and and we need to cure the patient using our knowledge. Yeah. So today we will act like a doctor, like scientists, but in virtual worlds. Yeah. In virtual reality, using this tools, using this metaverse. Yeah. Are you ready? Yes. Ready to answer the reality worlds? Yes. Let's begin. Okay. Now we still in still we still in mixed reality, and I will use here is Nova Science. Yeah, we will use these apps to enter the. Oh, I need to use the control. Okay, I'm sorry, we need new battery. And then could you help me please? Okay. Okay. Okay, now I'm in a virtual room. Okay, and we have the problem here, and this is the problem, and it goes here. Okay, first of all, I will teach you how to use it. Yeah, first how to pick. Yeah, to pick. Okay, thank you. To pick up, use this button. Yeah, use this button and for teleport. Yeah, to move to another place, use this button. And okay, sorry, my right hand still not connected. Okay, for every step, the instruction it will be on our left hand. Okay. So thank you ya. Control update in progress. Cancel update. Okay, now I have to answer. Okay, from here, I will give it to our class leader, Tati.
Okay. Ati, please, Ati, explain yourself.
Please lead your friend to solve this problem.
Okay. So, this is in a happy for we sell okay. So I'm going to proceed the process. So as we all know, the main goal here is to cure this patient, this cancer patient. How do we do that? We do that by creating a protein using the corrected gene. So in order to do that, there will be a lots of process, lots of steps that I'm going to be going through right here. So for the first step right here, we must identify the parts of the cells right here, which you already have in front of us right here, is the structures, the inside, internal structures of the cells, and we must determine which one of these names, these terms, fits in the specific structures that we're going to be needing for to create proceed our process in synthesizing the protein. Now I'm gonna call my friend right here, who is a professional, who is an expert in terms of cell studies. I'm going to call him from Salahuddin. Please come forward.
Salam, Masya Allah.
Okay, Salahuddin. I can't really see you, but can you tell me if you're there already? Okay. Okay. So, look, right here I have a lot of names right here, and I'm not really sure where to put these names. So, can you guide me on what should I do first in order for me to complete this step? And here, subject. See the lesson. You can take it first. Second, we'll try to take this. Okay. What should I put it? You can put it here. Here? This one? I think that's another. Oh, oh, yeah. Is it this one? Is it this one? Ah, okay. This one? Oh, yeah. So, Udin, can you explain to me a little bit of cytoplasm about it? So, what is the cytoplasm?
So the cytoplasm is like a fluid.
Oh, I see. Okay. So it's the fluid inside the cells.
Yeah.
Okay.
Forming the transportation of
Oh,
DNA.
so it's like a medium for the entire thing, the entire process inside the cell to
Yeah.
go on. Okay, let's move on to the next one. What's next?
Next is
Okay, I'm gonna get this one. Where am I gonna put this in?
you can put on the top the
This one? Okay.
Maybe someone try this.
Yeah. Okay.
Just try it. Okay,
You wanna try this one? Okay. The expert must use it to be more prepared.
the time is yet you should have an
Why you're here?
very
Okay. Okay.
I think this one is too easy, yeah. This is the one the cell structure.
Yeah, turn on. Yeah, okay.
It is headset battery is low. Yeah.
Now let's move on to the next process.
Yeah.
I think let's try nucleus.
Yeah. Yeah. So this is here, right? This one.
I think because nucleus is the center of the cell, center of the DNA and everything else. So I think it should be the center. Thank you.
Yeah, this one.
Yes.
Okay. Thank you.
It's like the entire point of the cell's existence depends on the nucleus. Right. Let's see what we got next week.
Okay. Let's try plasma membrane.
Okay. Which one?
Plasma membrane. Yeah. This is usually the plasma membrane.
I think because it's a membrane, it should be on the outer part of the cell.
This one. Yeah.
Yes, I think that's what it is. Okay, we're getting closer. The question of this cell membrane, the plasma membrane. What is it for? Is it for protecting the cell? Perhaps. Can you explain that?
So the plasma membrane is for the protecting the cell and for maintain the control and to maintain what the matter can enter the cell.
Oh.
Yeah. Or the cell.
Is it also for keeping the structure of the cell? Perhaps.
Yeah.
Maybe all of us. Okay. Let's see.
Next.
What's next?
Next, let's try this. That's what.
Okay, let's try that.
Is it ribosome? Ribosome.
Yes, that's your something.
So,
Oh, but then I noticed something.
oh, okay.
I see that on the rough endoplasmic reticulum
Yeah.
and this tiny part right here, there is it's the same thing, isn't it?
Oh,
So the rough endoplasmic reticulum also contains ribosome, is it not?
yeah. This is true.
Oh, I see.
It contains ribosome, but with different function.
Oh, can you explain a little bit about that function?
Okay, for this one is for creating for producing media. So I think it's different. How protein?
Oh,
Yeah. Producing protein. Yeah. And for the endoplasmic reticulum, this one is for producing lipid. Okay.
bro.
No, this one.
Or is it perhaps both are for produce protein, but for perhaps different usage maybe?
For different usage.
Ribosome and rope and the plastic reticulum both to synthesize protein synthesize and the difference is ribosome produce the ribosome lies in the cytoplasm to produce the protein or inside the cell. Root and the plastic reticulum or the nucleus can produce the protein for another cell, for another tissue or another things outside the cell. I see. Yeah. But then for our problem today, I think ribosome, protein, plastic reticulum, and nucleus will be useful to solve the problem detoxification, and what? Another one to for the cardio. I see you fit your business. Okay, and here it is. So we cleared up all this Microsoft stuff. Let's move on to the next one. Let's put the data. For the next one. Let's try it. So, this one. Yeah, I think should be the one. Difference between I think it's because nucleus is something that is inside, and the nucleus, the envelope one is something that is protecting as how envelope is. Envelope is something that covers whatever's inside it. So I think that's the same function for nucleus envelope. That's how it differs from nucleus. Okay, okay. So the next one is maybe mitochondria. Okay, let's start with the I think I'm a little confused about the site of mitochondria. Can you help me? Let me explain first. Yeah. I think mitochondria is this small red-looking thing. Yes. This one, right? This one? Yes, I think it's that one. Okay. Because as I remember from what I've learned, mitochondria is that's similar to how mitochondria is. It's small, it's an oval shaped structure inside it, and I don't think the blue one is one that is. Okay. But I think it must be the red one. Is it mitochondria for producing ATP or energy for our activities? Yes. According to what I've learned, mitochondria is powerhouse of the cell. So how does it become powerhouse of the cell? Is by providing the energy needed for the entire process inside the cell produces ATP. That's why it is the powerhouse of the cell. So maybe it's like nuclear, like electricity generator. Yes, like house. Yes, it provides electricity for the entire building. That's similar to how mitochondria provides energy for the cell. Okay, we got one left. Okay, maybe here. Okay, contacts. Again, you can see this. Yeah. Okay, but now I think please focus to the picture. The picture. Because it's it has something, so we can let's move to this side. Okay, so I think I can see the information from it. This is the picture, please. This is a picture. This is a picture. So okay. Please, focus to the picture. Okay, hold on. Okay. Okay, guys. As you can see from the rope and the plastic that you do. Okay, so please don't move. Sorry, yeah. Okay, there is from the rope and the plastic. Yeah, rope PR. Yeah, there are something small. Yeah, tiny. Consider this one, yeah. Move from the rope PR to Golgi complex, and then from Golgi complex there are many tiny things. You can move to membrane, cell membrane, yeah. This is like I told you before, yeah. This is to transfer the protein into the outside the cell, yeah. Okay. This is the cell structure, and we have completed. I think we can move to the next step. Okay, now that we've identified cell structures,
think we can move on to the next step. Can you take a look around and find something that could perhaps continue this process? Okay. Yes. Oh, I think they're in math and to my left and your left. Yeah. So take the bathroom and let's go to the top of yourself and pick up the empty. Oh, yeah, the scene. Okay. Let's see. Oh, that was. In order to support. Yeah. Yes. Yes. Okay. Let's put this. Turn around. Okay. Stop. Yes. Yes. Okay. Okay. Alright. Proceeding to the next step. Yes, Chinese. Okay. Next. Next person. Okay. What is that? See? And also breaking a glass. It's okay. Yeah. And then okay. Okay. Okay, so you want to use this method? Okay, you want to be using it. And then we'll be using it. Yes, for this process. The technician, the engineering technician. Okay. So, very cool. Oh, so is this? My hand. Wait, my hand. Pink is gone. Take the instruction first. Oh, yes. Take. I'm the left. But the left is go. Okay, we declare. Complete the sequence by placing the right nucleotides in the right order. Okay, so here we go. And see the sequence now. Now we have GVA. So C G. Okay, what we are doing here? We are connecting the transcription. We will transcript DNA from DNA to produce the protein. Yeah. Okay. 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 So what we're going to connect there? What is G connect to? G binds to C. C. G. What is C? What is C? Cytosine. What is T? T is timid. Connect with? Connect with yes. Ooh, it's moving. Oh, so each DNA sequences has its own connection. Yes, you're right. Oh, so when you just randomly connect it off. Okay, I have to go. So C, yeah, C, remember, baliknya lagi. Yeah, then what is change with? Not lipid. Yes. And then C. With G. With cytosine. Faster, guys, go back. We need to go faster, guys. Cytosine with A with C with adenine again. C uracil. Guanine, and then A uracil. Okay, it's moving. It's working. Sequence, yay! Wait for the RNA. Okay, we're waiting.
Okay, Nathan, want to try? Yes.
Oh, okay. I'm going to take this.
Handshake. Whoa. Let me go. Put it. I'm making it more faster. What is this? What is this? Okay. They're taking this. Where am I put it? Oh, here. Hey, kenapa? Sorry. Okay, so we're on the protein synthesis process in Netherlands. Are you going to finish it? Yes, so it is our next technician. Yes, this week. And of God. Mana? Victory. Yes, victory. You can continue my progress, victory. You can do it, victory. Yes, yes. And put it like this. Sorry. Somewhere we have process. Yeah. Okay. In cell, it should focus where? In yeah. What we should do is we have to translate the mRNA for making the whole chemical. Yeah. But not first the color. First, technically, you have to take the pattern first. The pattern. Yep.
Yeah, okay.
Okay.
It's for the ACU.
ACU, it should be A attached with U. It's for U. So the anticodon, it should be technical first. Yeah, I can my mind to speak English.
Okay, that's all from us. Shukran. Thank you for watching.
Okay, I'm sorry, I forgot to charge this device. Okay, it's done. Okay, in the last process, we need to complete the
Protein synthesis.
the translation, yeah, the translation. So we should to match between the codon and anticodon and amino acid. Okay, are you ready to matching it?
Yeah.
When you are ready, we will continue using this device. But hold on. Okay, Fatih, please lead the discussion here.
while I'm charging the device. Yeah.
Okay.
So we can prepare it better when we solve the problem. Okay. I think. Please.
Okay, hello guys. So while Ustad Agri is charging this awesome device, the VR, let's discuss about what we have completed. Yes. So, Fikri, can you perhaps conclude what we were doing in the first step while we were connecting the structures with the correct functions and stuff like that? What were we doing? Can you explain it? Perhaps summarize it and teach us something that we haven't known yet. The cell process, the first one.
Oh, okay.
Were you paying attention?
Yes. So paying attention. So the first one is transcription.
Okay.
So the cells will make a pair based on two chain, two like there are four interrelated with the minus of the and or the or the RNA. So it will explain here between A, G, A, and T or G and C. And if this process is done, it will change into the RNA,
Okay.
and the RNA move, will be walking onto the outside the nucleus through the cytoplasm, which is ribosome. And there's will be a process which we call this translation to make a protein.
Oh,
We started from AUG, called AUG, called basically as
a code. Something like a code.
codes. It consists of three nitrogenases.
Okay. So I think this correlates to how we're making a protein in order to cure this patient, right? Okay. So the correlation between protein and the nitrogen acid is nitrogen base. So like we have to connect this protein with the correct sequence of the nitrogen base in order for it to be giving the correct outcome. Yes.
Yes.
Okay, so I see. So Hata, can you perhaps what is it? Give us a little bit of a future vision on what are we going to do next after the process of what we should be using spring.
So I think I already explained the transcription step.
Okay.
And after that, after the last step of the transcription is when the RNA is go outside outside from the nucleus by the nucleus force, and then the RNA is attached to the ribosome, and it will become the next step is the translation.
The protein.
Yeah.
I see. Ejan, did you understand the entire process that our friends is explained and the entire practice using the VR that we have just done earlier?
Actually, from the explaining of my friends, I clearly just understand the substance. But from the thing that we do, the practical thing that we do, I understand the more topic entirely.
Okay, I see.
So because we do it in the same time, so we can know what the what do you call it? The bits or like the some amino stuff that do.
Okay, I see. So I think we can clearly conclude that by using this VR, by using this device, we can learn clearly and more easier. Correct?
Yeah.
Yeah. So do you guys think our class needs VR?
Yeah, I think it is.
Say it one more time.
Uh,
Degree,
Number one. The number one. Okay. That's one degree. I need to explore it. Okay, so we're going to look at the strand first. Turn around. Where's the strand? Oh, the we get the question first. Okay. Okay, so the start of the code is like always AUT, and then the next is UCG. Okay, so what's the next message friend? So this GU is no. Yeah, the ACU. So it will be UGA. UGA. Yeah. Is it correct? UGA. So now what's the amino acid? UGA. 1, 2, 3, 4, 5, 6, 7, 8, 3, 4, 5. 3, 4, 5. 1, 2, 3, 4, 6, 20. Yeah. So what's the amino acid? So that's the so the spread. The DNA spread. The DNA spread. No. That's this one. Yeah, this. It's good with you. Yeah, you. Okay. What's the C, K, U, okay. Yeah, that's right. Is it T? Is it T, U, G, U, will stop. It will stop. Is it stop? Yeah, will stop. Lah, dia isu. ACU, ATA. It's up. Is this up? It is. Okay, so we're using the correct. Okay, so the next one. U, U, G, U. So the you see, the couple is A, C, A, C, A. For them, for the U, G, U. A, C, A is G, U. Insane. DHR. DHR again. Okay, right. DHR, correct. So let's say that according to the mRNA stress. Yeah, the mRNA is according to the mRNA stress, not the RNA. So, which is HCA. The mRNA it's not DHR. Yeah, so you deal for C minus. Oh, cis. This. Okay, process. Yeah. Okay, next one. GCA. CGU. CGU. U. L. What? L. L. A. L. A. L. A. Yep. Right? Yes. Okay. So next one. Sorry. To like space. A U G U UAC. UAC. Okay. The amino acid. Okay, tell me. What is the AUC. 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 Right. AUC. 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 Man, use this. Really? Yeah. Okay, it's correct. I know. Methionine. Okay, it's okay. Okay. It's one. Between all correct. I'm waiting. Okay. So I could take this. So now we have polypeptide. Go to the next station to fold the protein. Okay. So where's the station? Oh, there. Yo, that's gonna go. Okay, so I put it here. Yeah. Yeah, it's still not. Press again. Press again. Okay. What is it? Still not. Still not. Yeah. I'm gonna shoot these to the outside. Cowabunga! Yeah, bye bye. Yeah. So we finished our protein synthesis. Yeah. Look at the text. Look at the text. The protein you produce. Okay, complex various chemical groups. There you go. Okay, stop. Okay, stop. Don't move. The protein you produce was modified and will be complex various chemical groups such as carbohydrates, phosphates were added, and the protein was further folded. And the final protein might be going complex version to move toward the plasma membrane. Once it reaches the plasma membrane, the protein was produced from the cell. This protein that you synthesize is coagulation factor that will cure the patient by restoring their blood capacity to coagulate. Congratulations, you cured the patient. Can you explain to us again? We had that again. Can you explain it to us? Yeah. Okay.
Protein produced was modified in the complex. So,
Can see the text.
produced was modified in the complex. So, after the ribosome,
Why focus in complex?
modified,
Yeah, modifying.
like adding some chemical groups
Like.
carbohydrates and phosphates,
Okay.
and protein is folded, more folded.
Okay.
And after that, the Golgi complex, the protein that in the Golgi complex was in another vesicle to move towards. So basically means like the transportation of the protein to move towards the plasma membrane.
Okay.
So when it reached the plasma membrane and the protein was released from the cell.
Okay.
And it's what is the factor that will patient.
So the protein will be what? Okay.
Coagulation.
Like that. And this one will cure. Relation. Yeah. Learning from this module is not just for this method. This is really helping to protect us. Okay, Alhamdulillah, we have done to our mission today.
success to cure patient. And yes, this is learning. This is the learning, and mashallah, yeah, you can apply your knowledge to solve the problem. Yeah. And mashallah, yeah, you can realize and make the good strategy to solve this problem. And so now we can move to the next step. Yeah. I want to check is the goal is achieved or not. Yeah. So I will check your knowledge again by test. So open again your modules, and there is a test. Please, please complete the test.
Okay. Finish? Finish, yeah? Alhamdulillah, yeah? Okay, so everyone has complete the test, and yeah, inshallah, I think this is all of you has the good result. Yeah, so today we have achieved our learning goals. Alhamdulillah. So before of that, I need to someone to give us the summary. Yeah, please summarize our learning today. Who want to be? Fikri, you want? Okay, inshallah. Come on, Fikri. Yeah.
So today we are learning about what we got today is about we know how is the protein synthesis works. Like that protein synthesis have the two stages, which are transcription and translation. And transcription is how the DNA is transferred onto the RNA, and the translation is how the RNA is exchanged to be carbon, which is a carbon will be making up to the specific carbon, and this specific carbon will make the protein. And we know that this process is essential because protein is one of the biological signal for our body, to how our body works. Based on how our protein is delivered, like the function.
I think by learning it using the QR technology, it makes it easier for us to understand the entire process, right?
Yes, I agree with that.
Yes, because by doing it, it's not that easy.
Yeah.
Very beneficial, essential for us.
With the virtual technology, we could learn something that we couldn't do like that in the daily life, in the real life. We could just do some surgery that, we still are children, we still are students, because we couldn't do the surgery because we have no what do you call it, experience about that.
Okay, thank you very much, and thank you, Fikri. Yeah. So alhamdulillah, we have done our activities today, and after this, I want you to feel the feedback. Give me the feedback. What you have success for today, and what you have not success for today, and how to improve for next week, so you can be better. And also give for me what is good and not good for me, and how I can help you to understand it more, to understand it better. And what do you like to for our activities next week? Because next week we will have the next topic. We will inheritance. So the next topic is about the inheritance. We will see how the genetic materials, how the DNA affect in our life. Okay, any question for today?
No.
Nothing? Okay, so I think it's done. Okay, you have complete your feedback?
Yes.
Okay, so nah alhamdulillah. Alhamdulillah ya rabbil alamin. Assalamualaikum warahmatullahi wabarakatuh. Oh yeah, after this we have Pasca KBM and I have uploaded the new assignment in our Google Classroom, right? Just check it and complete it later, yeah, in our Pasca KBM times. And don't forget for your KBM. See you later. Nice to meet you. Assalamualaikum warahmatullahi wabarakatuh. Jazakumullah khairan katsiran. Barakallahu fikum.